When solutes such as ions are placed in a solution without physical barriers, they become evenly distributed throughout the solution by a process called diffusion, the random movement of particles toward a state of equilibrium. For the mixture shown here, equilibrium is reached when the concentration of each ion is evenly distributed throughout the solution. Neurons, like all living cells, are surrounded by a plasma membrane that is impermeable to ions. This property allows a neuron to maintain different concentrations of ions between the inside and the outside of the cell by preventing the passive diffusion of ions from regions of high to low concentration. In a typical squid neuron, there is a large difference in the concentration of ions, such as sodium and potassium, between the intracellular and the extracellular environments. In addition, the interior of the cell has a high concentration of protein ions, each with a considerable negative charge. The plasma membrane is composed of a lipid bilayer. The hydrophobic interior of the bilayer prevents the movement of ions across the membrane. The only way that ions can diffuse across the membrane is by passing through specialized channels that permit the movement of particular ions while excluding others. These channels can be in an open or closed state. When the neuron is at rest, most ion channels are closed. However, a few potassium channels are open, and potassium ions are therefore free to diffuse out of the cell into the extracellular environment. Note that channels can be highly selective. The potassium channels do not permit the passage of sodium ions. Since only a few sodium channels are open at rest, very few sodium ions flow into the interior of the neuron. If we consider only the potassium ions, which are relatively free to cross the membrane, we notice that the intracellular concentration remains much higher than the extracellular concentration. This is because the potassium ions are pulled by two different and opposing forces. First, a diffusion force acts to pull potassium ions down their concentration gradient toward the exterior of the cell. The movement of potassium ions out of the cell results in a depletion of the internal positive charges, causing the potential to become increasingly negative. An electrical force, produced by attraction of the positively charged potassium ions toward the negative cell interior, generates an electrical gradient that pulls potassium back into the interior of the cell. The diffusion and electrical forces eventually come into balance, and an electrical potential is achieved at which the electrical gradient is exactly balanced by the diffusion gradient. This potential is the equilibrium potential for potassium. At this electrochemical equilibrium, there is no net movement of potassium ions. On average, for each potassium ion that leaves the cell, another returns. The electrical potential across the membrane can be measured using a pair of electrodes. An oscilloscope is used to continuously display the potential difference between the reference electrode and the recording electrode. When both electrodes are in the bath outside of the cell, no difference in potential is measured between them, and the display registers 0 millivolts. However, when the recording electrode enters the cell, the display records a membrane potential of about -60 millivolts. This value represents the resting membrane potential of the neuron, the difference in electrical potential across the membrane during an inactive period. Communication between neurons and communication between neurons and muscle occurs at specialized junctions called synapses. The most common type of synapse is the chemical synapse. Here, we examine the events that take place at the neuromuscular junction, a chemical synapse using the neurotransmitter acetylcholine. Synaptic transmission begins when the nerve impulse reaches the presynaptic axon terminal. Depolarization of the presynaptic membrane initiates the sequence of events leading to transmitter release and activation of receptors on the postsynaptic membrane. Shown here are the principal molecules and organelles necessary for release of neurotransmitter. Synaptic vesicles exist in distinct pools, either tethered to the cytoskeleton in a reserve pool or free in the cytoplasm. Some of the free vesicles make their way to the plasma membrane and dock as a series of priming reactions prepares the vesicular and plasma membranes for fusion. The membranes of the synaptic vesicles are drawn together via protein complexes, collectively called snares, that are expressed on the vesicle and presynaptic membrane. When the axon terminal is depolarized, voltage-gated calcium channels open, and calcium ions rush into the axon terminal. Some of the calcium ions bind to a protein on the synaptic vesicle membrane called synaptotagmin. When calcium binds to synaptotagmin on the synaptic vesicles nearest the active zone, the vesicles are drawn even closer to the presynaptic membrane. The vesicles fuse with the axon terminal membrane and release their transmitter cargo into the synaptic cleft. Some of the transmitter molecules bind to special receptor molecules in the postsynaptic membrane. The response of the postsynaptic cell, either excitation or inhibition, depends upon the particular neurotransmitter and receptor combination. For example, the receptor for acetylcholine is permeable to sodium. After binding acetylcholine, the channel opens and sodium ions enter the postsynaptic cell, thereby generating an excitatory postsynaptic response. Transmitters are inactivated or removed rapidly from the synaptic cleft so that transmission is brief and accurately follows the presynaptic input signal. For acetylcholine, an enzyme in the synaptic cleft, acetylcholinesterase, breaks down acetylcholine into choline and acetate. The release of transmitter from the receptors causes the channels to close. Not all transmitters are broken down by enzymes in the synaptic cleft. Many transmitters are rapidly cleared from the synaptic cleft by being taken up into the presynaptic terminal by special proteins called transporters. This process is known as reuptake. Reuptake not only cuts off the synaptic activity promptly, but also allows the terminal to recycle transmitter molecules. The membrane needed for creating synaptic vesicles is also recycled via endocytosis of the presynaptic membrane. The recycled vesicles are refilled with neurotransmitter molecules and are ready for another round of synaptic transmission. The indoleamine serotonin is synthesized from the amino acid tryptophan in two chemical steps. The enzyme tryptophan hydroxylase adds a hydroxyl group to the amino acid to form 5-hydroxytryptophan. The enzyme aromatic L amino acid decarboxylase then removes a carboxyl group from the molecule to yield 5-hydroxytryptamine. Because its chemical name is 5-hydroxytryptamine, serotonin is abbreviated 5-HT. Serotonergic cell bodies are relatively few and are concentrated in the raphe nuclei of the midbrain and brainstem along the midline. Large areas of the brain are innervated by serotonergic fibers. Serotonin has been implicated in the control of sleep and wakefulness, mood, anxiety, and many other functions. Termination of the effects of synaptically released serotonin occurs largely by means of a reuptake process. Cytoplasmic serotonin is also metabolized by the enzyme monoamine oxidase, or MAO. A large number of serotonin receptors have been identified. All but one are metabotropic receptors. A number of drugs have been found that stimulate 5-HT release, for example, fenfluramine and many amphetamines, and thus act as potent agonists. Drugs that inhibit the reuptake of serotonin, the so-called selective serotonin reuptake inhibitors, or SSRIs, are often used to treat depression, anxiety, and obsessive-compulsive disorder. These drugs include fluoxetine, Prozac, clomipramine, anafranil, and sertraline, Zoloft.